We are here for the Global Cannabis Research Summit online and Section 2. This is an innovator from Denmark, Octarine Bio, and I won't say much other than I'm excited about learning more about the topic being, biosynthesis. And these are involving targeted cannabinoids, but it's not the only thing that's on Dr. Gallage's mind because of her background and where her expertise comes from. So welcome, Nethaji.
Thanks, Keith. Thanks for having me here today.
Very excited about the presentation, and I really like this title slide because of that intense, turbulent sky that might be the market, in fact, that is cannabis as it emerges. So tell us about your startup and why you're excited about what you're doing these days with Octarine out of Denmark.
Thanks, Keith. And as said, thank you for the introduction. Also, my name is Nethaji, and I'm the founder and the CEO of Octarine. Would like to actually give a background myself before I start, guide you through our technology at Octarine. Since this is a research summit, I'm taking the liberty to talk science here. So that also because I'm a scientist by training. I have been part of University of Copenhagen Center for Synthetic Biology, where I have had my own research group working on cannabinoid biosynthesis. I'm specialized in elucidating plant pathways for high-value molecules and then engineering those in microorganisms to have sustainable production systems. In my previous research, I have been able to engineer vanillin flavor in microorganisms, and this was quite successful and became a commercial product. I also have background in business development in the industry as well as research in the industry. So that's how Octarine came about. So we are based in Copenhagen, and to your right side in dark green, you can see where we are located, close to Norway and Sweden. We are a spin-out from University of Copenhagen and Technical University of Denmark. We are quite early stage, started up in 2018, and then closed our pre-seed round early this year, together with Danish government venture arm, Vækstfonden, and a syndicate of business angels, including Bruce Langdon. Most of you know who he is. He's the former CEO and the founder of Canopy Growth Corporation. But just to tell you why we are here, why we are interested in these molecules, it is clear to us that many neurological and psychological conditions are poorly served by current drugs and leaving physicians, patients, and their families desperately looking for alternative therapies, and even sometimes illegally. Cannabinoids and psychedelics are poised to offer breakthrough therapies for these conditions, and to realize full potential of these molecules, issues with their production and undesirable properties of these molecules has to be solved. And that leads me to my next slide, why we are here with our technology. For us, it is clear that there are two key problems in the industry today. One is the problem with the production, and the other problem is actually with products. Today, the production is almost entirely based on plant extraction, which is extremely resource intensive, and some of the rare cannabinoids that are actually showing very good pharmaceutical profiles are present in very low levels in the plant. It is also clear that the quality of plant extracts varies each time, making them unsuitable for various applications. And if you are aware with the FDA guidelines that came out recently, the quality and the pure molecules are going to be a key thing in the future. The second way to produce these molecules is by chemical synthesis. It is still very costly, and the iteration from one to another molecule is very slow. This is just one side to the problem. So the other side is actually natural molecules have poor properties in various applications. We know that these molecules are having very low bioavailability in human, and they are incredibly insoluble in water. And some molecules are psychoactive, which is also undesired property in many applications. And poor absorption is also a problem in certain applications. And one of the things that is not in this slide is the chemical instability of these molecules. Our solution is a biosynthetic platform that can produce both natural and improved cannabinoids. We use engineered yeast to produce these molecules and combine that with the enzymatic platform in order to improve them. This combined technology gives us a cost-efficient, scalable, and stable supply, and 99% pure molecules, and access to otherwise inaccessible novel molecules, which expand the chemical diversity within this class of molecules. And these improved molecules are actually patentable. So, bringing you more deep dive to the biosynthesis of cannabinoids in microorganisms, it is very much simplified in this slide. What we basically do is to copy the plant machinery into the yeast. And here in this slide, it's visualized putting A, B, C, D together into the yeast, and then you do the brewery of these molecules. But actually, in reality, it's rather complex. To make you understand the complexity of these biosynthetic cannabinoids, I would like to deep dive into the plant production of these molecules. It is clear today that cannabinoids are stored in glandular trichomes in plants, especially in flowers. They are not only present in trichomes, they are actually present in the oil bodies of trichomes, which is nicely visualized in the picture. In these oil bodies, it's not only one cannabinoid. There are 100, 130, or even more different cannabinoids molecules are sitting there as a chemical cocktail. And why do they do that? Why do plants have this chemical cocktail sitting in their trichomes? One of the reasons that it is believed that these are plant defense molecules against pathogen. And it makes sense that you have a chemical defense cocktail, and when the pathogen hit by these cocktails of chemicals, they have difficult time adapting to these because there are many molecules with similar structures. And that is quite clever for the plant, but for us who are bioengineers and every other biologist, it's still complex to understand how plant producing these molecules. Even though they are stored in these trichomes, it's not the place that they are synthesized. These molecules are synthesized in different tissues and even in different organelles within the cell. So in this case, actually, devil is in the details. So my research group at University of Copenhagen for 3 years studied the plant biosynthetic pathway for cannabinoids. What we have done was to profile different proteins in different tissues and also the genes encoding these enzymes. That is the one way to understand each different tissue, what's going on in there, and then put this puzzle together. To your right side, there is a figure showing the chemical biosynthetic pathway or the biosynthetic pathway of cannabinoids in cannabis sativa. And I don't want you to focus on all the structures and all these different enzymatic names. Just want to visualize you how complex this is. It's coming through amino acid, and there are multiple steps converting intermediates all the way to CBGA, THCA, whatever other cannabinoids. And even in this figure, that it's very much simplified. You can see certain places there are multiple arrows showing different steps. And luckily, before us, there have been research groups understanding different steps, but one of the steps that was not known until very recently, actually until this year, was one of actually the key step of plant biosynthetic pathway. And that's the step where cannabidiolyc acid is made. And the enzyme converting this catalytic step was unknown for many, many, many years. A research group from Berkeley, USA, and research group from my group have been neck to neck finding out this research showing actually which enzyme that is behind this reaction. And to our surprise, this actually enzyme also sitting in a different organelle in the cell, which is the chloroplast. Chloroplast is the photosynthetic unit in the plant. Photosynthesis is where the sunlight and CO2 become energy. And that's where one of the key steps of cannabinoid biosynthesis is taking place. Just want to make sure that you understand the complexity here when you have to engineer such pathways in microorganisms. It is still not known whether this enzyme is sitting in the inner or the outer part of the chloroplast, and actually why that enzyme is sitting there, whether it is the composition of chloroplast, whether it is because it's near to the energy cells. There are many things that here we are still not fully aware of. With that, I'm going further into how that you elucidate biosynthetic pathways. And in my previous slide, I told you that we do this proteomic genomic analysis, and then we gene mine each different step. But these enzymes has to assay in different organisms to understand if they actually have this catalytic activity. One of the way to do it is to express this enzyme in a different host. We use, in our case, tobacco plants, but please be aware the use of tobacco plant has nothing to do with producing cannabinoids in tobacco or anything like that. Tobacco is actually model organisms to test various enzymes. This has been used for decades in academia. Tobacco is easy to manipulate, and there's a lot of molecular biology tools to do so. So what we did was to assay these enzymes in tobacco to see that they have these activities. We found very quickly that tobacco and any other plants actually so clever at understanding something wrong is going on when something new is expressed. So they all the time breaking down whatever new products that we are producing. So if the cannabidiolyc acid is made, then it's been converted further. So this gave us some indication actually having a higher organisms to establish a biosynthetic pathway is going to be rather challenging, because the challenge is not only assembling a pathway, but also keeping your product safe in there without being broken down. So it would rather add another layer of complexity of using plant or any other higher organisms. So we choose to work with yeast. Yeast is a simple unicellular organism, and it has a very short life, so the fermentation cycles are much quicker, and it doesn't do this breaking down of molecules the similar way as plant does, so you get your product. The purification process is quite easier as well, and there are quite a lot of molecular tools to work here because it has been used as a model organism for many decades. So it's not only cannabinoids that we are interested in producing through synthetic biology and bioengineering. We're also interested in producing psychedelics. So during last 1.5 years, my co-founder Nick has been able to engineer yeast to produce psychedelics such as psilocybin. And to our great surprise, we have been able to achieve 1 gram per liter at proof of concept stage, which is a huge yield considering how difficult engineering process is in other organisms. But one of the advantage here is copying a pathway from fungi to yeast is much more straightforward compared to, as I tried to clarify previously, copying a pathway from plant to yeast, especially cannabinoid biosynthesis. But as we are yeast engineers. I think it's not coming as a huge surprise. As we are based in Copenhagen, Denmark, yeast engineering is, I think, written behind our back. There are quite a few very big pharmaceutical and CPG companies based here working with fermentation and yeast technology. So the whole infrastructure is quite available for us. And then there is also access to very much highly skilled a workforce who are specialized in yeast engineering and fermentation in Copenhagen. That gives us quite a big advantage in this industry. And to those who actually have not heard about biosynthesis and bioengineering products, I can say that this is not a new technology. The technology has been here for almost 2 decades now, and there are products in the market already. There are flavors, fragrances, pharmaceutical products, and other products already out there for the consumers and patients that are provided through these technologies. What is new is to use the same technology for cannabinoids, which is an extremely complex biosynthetic pathway. Second part of my talk is going to focus on how we're going to improve these molecules and what we do at Octarine to achieve this. As I mentioned previously, we are working with enzymatic modification platforms, which is called in-cell enzymatic platforms. One of our platforms that I'm going to introduce here is our glycosylation platform, which has more than 380 enzymes. The glycosylation is an enzymatic reaction where you can add various sugars to a small molecule. So the sugars can be added to various positions targetedly or untargetedly. These sugars could be glucose, xylose, and any other sugars and can be added in combination. Having such a comprehensive platform, we have quite a lot of variation to play here. Glycosylation is a concept that has been there for quite some time, and actually this is something that University of Copenhagen and my research group has been pioneers on, especially my PI as one of the first to actually use this technology in commercial and industry scale. So it's not a surprise that we are quite interested in the same technology, which we have very much hands-on experience working in. And when we have applied this to cannabinoids, we found that actually, indeed, that we can improve the quality of these molecules rapidly in a very, very good manner. So when we run the cannabinoids through our enzymatic platform, we modify them in a certain way. As I mentioned, this could be a stable modification or intermediate modification. But then we send these molecules for in vitro screening cascade where we validate actually the improvements. So today we have a collection of molecules that are extremely soluble and more bioavailable and chemically stable, etc. So yeah, as I mentioned, some of our molecules are very much water soluble, incredibly water soluble, I would say. As you can see in the picture, they are completely transparent, odorless, and completely solubilized in water. We have different molecules that are extremely stable under extreme condition as extreme pH, temperatures, etc. And they are resistant to oxidation. You would not see the purple color forming if you have these CBD molecules. We also don't see any degradation of THC or the CBD to THC, which sometimes happen with the CBD molecules. As I said, that we can also increase the bioavailability. We can see from the in vitro profile data that we have molecules that are resistant to liver detoxification and have shown improvement of intake in bloodstream. So how are we going to translate these technologies and our novel molecules in applications? It's clear to us that our main focus is going to be pharma because of this incredible profile they're already starting to show. And one of the places we would be targeting too is drug-resistant epilepsy, where we already know the current drugs can be much more improved if you look at their bioavailability and oral dosage and so. These molecules can also be used in pain treatment, where the poor absorption is quite a big problem. And with our enzymatic modification, we can also remove the psychedelic properties of certain molecules such as THC. There are also applications in consumer industry, and we have been approached by several companies, especially in the beverage industry. So in this slide, you can see our pipeline. We are rather far with our cannabinoid glycoside and pipeline for preclinical. We have done the validation and moving toward preclinical studies with these lead molecules. We have proof of concept production strains for producing natural cannabinoids as well as psychedelics, not only psilocybin, DMT, TMT type of molecules as well, and we continuously expanding the psychedelic production strain to others. So I am very much lucky to have a very skillful team behind me, and very lucky to have my co-founder Nick with me in this journey. He's coming from very strong bioengineering and fermentation background. He has been a researcher at Technical University of Denmark and previously also worked for biotech companies. And I have hands-on experience in commercial projects, upscaling similar projects as engineering, cannabinoids, and psychedelics. I have my CBO, Christian Tellgren, who's coming from Novozymes background with many years of business development experience. We have Christian Hansen, our CTO, more than 20 years of experience from drug development at Novo Nordisk, and some very skillful research scientists from University of Copenhagen and other Danish universities. We have Morten, who's our head of IP, who's excellent in synthetic biology IP landscapes. I'm lucky to have quite a few key opinion leaders who are coming from scientific background. Kim Anderson, who's the former senior vice president for Lundbeck. John Louis, coming from Pfizer for many years in medical chemistry. Bjørn Bemola, my former PI, is a guru of plant biochemistry, which is also quite important for us right now. Yeah, with that, I'm going to leave it there.
Impressive team, impressive story. Dr. Gallage, really, thank you for spending time. And just to wrap up with some questions, actually, I thought Gaetano online had some good ones to talk about. You know, as far as, I think you covered some of this, but as you think about scale up, where do you think, you know, from a competitive standpoint, when we talk about putting aside superior, you know, qualities that might even demand a premium price, right? But in a competitive head-to-head situation now, how far do you imagine you need to go to make it competitive, or already are you there from a material standpoint?
Yeah, so when we talk about the competitive or in relation to cost of goods, then we have to talk about which industry we are talking about. Because plant extraction, cannabinoids to consumer market where the quality doesn't really matter, that's actually a difficult place for us to compete. Then we would need much higher yields. But that's not where our biggest value proposition is. It's these high quality molecules that are needing for medical and pharmaceutical purposes. And that's where our target is. And with the biosynthetic natural cannabinoids, I would say 3 to 4 years of timeline is what we're looking at.
And do you see the production or early stage, the next stage efforts being close to home in Denmark or through licensing? Do you see this going global pretty fast? How do you feel the next year will unfold for Octarine Bio?
Yeah, I mean, I am quite surprised by approach by European B2B companies and even Australian and Asians. It seems that there is opening up the field and the industry other places than US and Canada. And these business or commercial partners have reached us for collaboration in order to get hands-on biosynthetic cannabinoids with high purity.
And what kind of industries or applications can you mention, even not names, but more sort of general? Are they all in combinations of CBD and THC for specific kind of areas, or dosing, delivery? What would you say of the current mix?
Some of them are interested in natural CBD and THC products for the medicinal industry still, not the consumer. But others actually are quite interested in these new molecules with improved properties for different pharmaceutical applications. So it's a combination of pharma companies and then companies who have focused on both consumer and medicinal market, but want to specialize in the medicinal market.
And I saw on your picture before, and it's always good to have a slide with pictures. That's what I remember from applications in the examples. I saw a transdermal type patch delivery. I would imagine this is, again, a potential in the mix with what we heard earlier from Psychedelics, right? You know, in terms of there's the technologies that complement what you're actually developing is maybe the active ingredient, if you will, and that was more of a delivery mechanism, right? You know, technology. So are you finding collaboration or potential collaborations? You mentioned Australia and other countries, but sort of innovators sort of connecting with you to sort of connect the dots, so to speak, bring what they bring and try to combine. Anything interesting you can tell about that?
I could certainly say that we are quite open for collaborations and in dialogue with many partners. I think in this industry, the collaboration is the way to move forward. There are a lot of different third parties with different specialities and competencies. I think we are much stronger if we can establish these collaborations. So we are quite open to that, definitely. I'm afraid I cannot disclose any names right now. And the details, obviously, we are also in touch with.
What about any potential barriers? I'm just thinking out loud. If you take California, an adult use and medical marijuana market, will the product or material, right, you know, that you're producing have to go through, you imagine, a different route of approval? Has that started to develop in places like Canada? What would that look like for this type of biosynthesis vector?
Yeah, it's again different according to where you want to develop your products to. So our improved cannabinoids with this very good in vitro profile with improvements that we are looking for the preclinicals, that's definitely because we want to enter this pharmaceutical approach, going through the same as any other pharmaceuticals.
Full pipeline. Mm-hmm.
Full pipeline, exactly. But regarding any other application like medicinal over-the-counter, as I said, biosynthetic products are not new. If it is the natural molecules that are made, like CBD and THC, then the regulation process is actually quite straightforward because there has been products approved to the market before. So the infrastructure is there, very well established, both in Europe and in Canada and in the US.
And I give credit to where you are home based in Denmark for actually, you know, in their pilot effort to create a regulatory, right, policy framework that allows you to make those connections, import, export. It's early days, but it's the right direction. Thank you so much, and I wish you well. I know we had you join past dinner time, and you have a family, so well, we really will take this information, and it's going to be combined with a lot of things we're learning about. But Dr. Gallage, wish you all the best, and again, thank you. We're going to move on to our next presentation. And thank you very much.